Ladies and gentlemen, good evening. My name is John McGraw, and I am the Interim Vice President for Research and Economic Development here at UNM, and I truly do welcome you to a very important, very significant, and very happy event for UNM. Thanks for joining me this evening for the University of New Mexico's 58th Annual Research Lecture. Tonight, we celebrate the significant achievements of Associate Dean for Research, Scott Burchiel, from the College of Pharmacy, and to present him with this year's Annual Research Lecture Award, which is the highest honor for research and creativity that UNM faculty can bestow on a colleague. The University of New Mexico places high value on research and prides itself as being designated a very high research university by the Carnegie Foundation, a designation specifically for universities that actively engage in research over a broad range of disciplines and fields. Last year, UNM faculty researchers generated more than $125 million in external awards in support of UNM's research venture. This amount represents but one aspect of our faculty's commitment to research and is illustrative of their commitment to excellence. UNM faculty success in securing these awards provides the university with critical resources necessary to enhance the quality of teaching and student participation in research efforts. It is important to note that these research funds provide valuable opportunities for all of our students, graduate students, undergraduate students, and we allow them to engage in cutting-edge research with national and international exposure and a wealth of experience that will literally contribute to their future success. We also recognize that our faculty lead the way towards these endeavors, and tonight I would like to take the opportunity to personally thank Professor Burchiel for his contributions and leadership in this effort. We look forward to his lecture on toxicogenomic bases for human susceptibility to environmental injury and disease. It is now my pleasure to introduce Linda Wellage, who is Professor and Dean of the UNM College of Pharmacy, who will be giving introductory remarks regarding our award recipient, Dr. Burchiel. Linda has been Dean of the College of Pharmacy at UNM for approximately 1.5 years, where she works closely with Dr. Burchiel in his role as a faculty member and Associate Dean for Research. Prior to joining UNM, Dean Wellage was Professor and Associate Dean for Education at the University of Michigan College of Pharmacy, a translational clinical scientist, and a director of the Education and Mentoring Core in the Michigan Institute for Clinical and Health Research. Following Professor Burchiel's lecture, there will be a brief question and answer period, and then Provost and Executive Vice President for Academic Affairs, Chowki Abdallah, will do the honor of presenting the award to Professor Burchiel. Please join me in welcoming Dean Wellage. Thanks.
I am honored to introduce Dr. Scott Burchiel as the recipient of the University of New Mexico 58th Annual Research Lecture Award. As mentioned, one of the highest honors the university bestows to a faculty member. Dr. Burchiel joined the University of New Mexico in 1977, first as a National Cancer Institute NRSA Fellow, and then became a faculty member in the College of Pharmacy in 1978. Dr. Burchiel is the Nunzio and Sherilyn DeSantis Endowed Chair of Pharmacogenomics and Professor of Pharmacology and Toxicology in the Department of Pharmaceutical Sciences in the UNM College of Pharmacy. He also serves there, as you heard, as the Associate Dean for Research and directs the New Mexico Center for Isotopes in Medicine. Dr. Burchiel is internationally renowned scientist whose career exemplifies outstanding achievement in research, education, and leadership with a lasting impact on the university and on the field of toxicology. Dr. Burchiel has been instrumental in developing and supporting the graduate toxicology program in the UNM College of Pharmacy, which has been active since 1988, and in promoting toxicology education throughout the Southwest region based on his tireless contributions and leadership within the Mountain West Society of Toxicology. He has been the principal advisor and mentor for 15 graduate students and 12 postdoctoral fellows. Dr. Burchiel is tremendously committed to strengthening environmental health sciences and preparing the next generation of leaders in this field. Throughout his career, Dr. Burchiel has been at the forefront of environmental health research. He was early to recognize that environmental health studies needed to expand beyond heavy metal and smoke exposure to more genetically based studies that can lead to predicting how a particular person may respond to a given exposure. He is an international expert in immunotoxicology and environmental carcinogenesis, who has been continually funded by the NIH for more than 25 years. Dr. Burchiel's laboratory is active in the use of toxicogenomics, pharmacogenomics, and the use of genomic and epigenomic tools to analyze mechanisms of action of chemical compounds. These studies seek to identify human susceptibility factors, such as polymorphisms in receptor signaling, metabolism, and in DNA repair genes. Dr. Burchiel's research leadership has also transformed this institution and the field of toxicology. In particular, he has shown a gift for building collaborative interactions that are the current paradigm for scientific innovation and progress. To paraphrase Dr. Dieter of Cornell University, Scott sees large-scale research, jigsaw puzzles, and provides those missing pieces. It is so incredibly valuable. It is what Scott has done time and time again over the decades. His ability to reach out and form partnerships, including those with other universities, those with Loveless Respiratory Research Institute, and our national labs, has been invaluable to stimulating the growth of the university's environmental health programs. His leadership was in part the reason the university was able to obtain the NIH-funded Environmental Health Center grant, which he directed for more than 7 years. Dr. Burchiel also served as the director of the UNM Health Science Center Signature Program in Environmental Health Sciences and as an associate vice president for advanced studies. He is currently a fellow of the Academy of Toxicologic Sciences, is an active NIH grant reviewer, is the editor-in-chief of the Toxicology and Applied Pharmacology Journal. He currently chairs the FDA's National Center for Toxicologic Research Science Advisory Board. And I could go on and on. But in addition to those impressive accomplishments, I want to personally say, I know that more is yet to come. He was recently funded to study the effects of arsenic and PHA on the immune system and has partnerships with the University of Arizona, the University of Oregon, and with Cornell to further propel the field of environmental toxicology forward. But prior to introducing Dr. Burchiel, I would also like to recognize his lovely wife, Judy, and his sister, Christine, who have supported him throughout the years. And it's now my pleasure to introduce Dr. Scott Burchiel as the University of New Mexico's 58th research lecturer, whose presentation tonight is entitled Toxicogenomics, Basis for Human Susceptibility to Environmental Injury and Disease. Please join me in welcoming Dr. Burchiel.
So I'll ask somebody to come reboot the computer so we have slides here in a minute. It's just a great pleasure to be here tonight, and I thank you for your attendance. And I have so many personal friends and people who have come back to UNM that have been people I've worked with for many years. You know, as you were reading my introduction, the university requires us to write something about ourselves every year we're here. So she had 36 and a half lines. That's something you have to fill out for the annual update to your biographical record. And so that, you accumulate things over the years. But it's been just such a great environment here. I'm so appreciative of the university. I'll have more to say about that as I get into my talk a little bit. But I just want to thank you very deeply personally for coming, so many friends that have come tonight and come back to the university. The title of my talk, which was on one of the slides we're going to bring up here in a second, Toxicogenomics. Now, this is not too big of a group. This is about the size of my lecture group in pharmacy, which we done for many years. I teach pharmacology and immunology and toxicology to graduate students. And it's about the size of our PharmD class. So we could keep it a little bit informal. The way we would normally start off an evening like tonight is, or not evening tonight, but a lecture, I should say, is everyone would have an iClicker in their hand, and an iClicker is an immediate electronic feedback. And that feedback is I can ask a question, and then you can pick A, B, C, D, E on the screen. And then I know kind of what you're thinking, or if I'm getting through. One of the things that I like very much about tonight is that we could you go ahead and boot the PowerPoint, please? One of the things that I like about tonight is that they said it's a lecture and not a research seminar. I'm pretty accustomed to going on the road with my slides and giving this talk. In fact, I'm going to give a few of the slides from the end of this talk at a meeting I'm going to in a few weeks. And so my goal today is to bring you up to speed to tell you kind of some of the things we're doing in the lab today, because these are the things that excite me and keep me interested in this research. So I just wanted to ask for a little bit of feedback. We did not get the iClickers out this evening, but I want to know how many of you know what the word toxicogenomic means? If you put okay, Matt, come up here and explain it to him. So my colleagues from the College of Pharmacy, thank you very much. Actually, our students know, and there are a few students here tonight, and I thank you for coming. Our students know. In fact, the first part of my talk, they could give the lecture. So I just want to say that what I'd like to do tonight is, first of all, start off by thanking the students that have worked in my lab over the years. And there have been many of them. And I could tell, as obviously, a story about each one of them, but I don't have time to do that. And I won't chase those rabbits, as I told my dean today. Some of the people are here in the room tonight. May Fen is here. I reckon, were you May Fen? There she is. She's waved her hand, but she's very shy. And most of these people have gone on to careers in academia or industry. In fact, one of the people who doesn't officially show up on my list is Todd Thompson. Where's Todd? And Todd actually is pretty much responsible for organizing my nomination in secret, I might say, that I didn't know that they were conspiring to get people to write letters and to support my nomination this year, and I do appreciate that. But Todd was in my lab for a number of years, and as his wife is now, Deborah McKenzie, is in my lab now as a research faculty member. But it's people like Todd and Deborah that make us want to come to work every day. So these are a list of my graduate students. I'm actually saying thank you. I really can't see the slides that well from here, so I'm like this. It's like you're in New York City trying to see up. But thank you, graduate students, and Others, June Gao is here. I'm going to actually, June, where are you? Thank you, June, for coming. June now is a staff member at Los Alamos. And I just really appreciate the work that these people have done, and I'm going to highlight some of their work. It is their work, not my work, that we're going to be talking about tonight. I just get to say, go to the lab, bring me data, and let's see if it looks good, if we can make a story out of it. Let's see where the gaps are, and then let's try to get it published. These are postdocs that we've had over the years, and I know that Matt Reed, sitting in the front row, is always there for me, and I appreciate that. John Davis was here last week with his family, but couldn't come out tonight. He actually is in Cambridge, in Boston, and others that we've had over the years. But I want to tell you, I picked one area to talk about tonight. I mean, I work in several areas, but I wanted to talk about genomics tonight. And the reason is because I've received this endowed chair from Nuncio DeSantis. I don't actually know, it was 8 years ago, I believe it was. And it has been the greatest opportunity, the greatest freedom I've ever had, is to have an endowed chair where we can take the funds from that money. And if you don't know what that means, it means they give money, and in the year that it was given, the state matched it. So there was a sizable amount of money, and then I get to use the interest. I don't get to spend the corpus. So when interest rates are good, or investments are good for the university, I have a great year in the lab. They haven't been that great the last few years, actually. But we managed to save the money, and we managed to use it for things that we have a passion for, something we really have an interest in. So I know what I'm going to talk about tonight is a little bit technical. My sister said, "What is the genome?" And I was like, okay, I'm in trouble. So, but I appreciate her. I love her, and I thank you for saying that, because it keeps me reminding that not everybody does this stuff every day like I do. So what am I going to talk about today? I'm going to talk about tonight. This is basically an outline, and I'm going to talk a little bit about introduction to pharmacogenomics, or we have abbreviations for everything. And so I won't use that too much, but people use that, PGX. I'll talk about toxicogenomics, which really are the same thing. And I'll be talking about, what they are. And one refers to a pharmacologic effect, maybe a drug or an intended effect. Well, toxic sounds bad, right? So it's usually an unintended or an adverse effect. And that's what we're going to talk about, the genetic basis of some of these chemical effects that we experience. And I'm going to start with things that are comfortable to you, and then I'm going to make you very uncomfortable, talking about things in the environment, things that you don't think about or worry about. So that's where we're going to go. And then I'm going to use as an example hypersensitivity reactions to drugs and foods. I know many, and I'll find out. I'll do a little quiz here in a little bit, or a query, and ask who's sensitive to what, because it's actually much more common than you would think. I'll then talk about what I do, environmental health. So I'm going to then talk about environmental chemicals, things that we're exposed to in our urban environments or in our drinking water, et cetera. And I'm going to try and introduce a difficult concept to you, and I'll talk about, staying on the hypersensitivity or allergy type theme, I'll talk about beryllium. And this is a problem that we deal with in New Mexico at Sandia and Los Alamos, because workers work with beryllium, and I'll tell you a little bit about beryllium sensitivity. And then I'm going to really get into my own research, which is environmental agents which suppress the immune system. And I'll talk about, this will be the hard part of my talk for you, but I want to give you sort of an appreciation of the genes that are impacted by these chemicals. And so I'm going to talk you through a few lists of genes and try and get you, I don't think you'll be comfortable, but at least you can appreciate that these chemicals are doing interesting things in your body, and we need to understand them. And then finally, one of the things I've always been interested in is technology. And I really, it's been amazing. I mean, everyone today with their iPads and their iPhones, they realize how much things have changed since we were undergraduates or graduate students. The personal computers, the World Wide Web has totally changed our world in terms of how we ask questions, what kind of research we do. So I'm going to show you some examples of things that are hopefully going to blow your mind, because it's just amazing the kinds of tools that we have today. We can ask big questions. We can ask audacious questions, and that's fun to be able to do that. So here's Pharmacology 101. And I want to thank the interpreters tonight for doing this. My daughter's an interpreter and studied sign language for many years, so I really appreciate the effort to communicate with everyone. This is Pharmacology 101. Now I could hand it over to, I could call one or two out of the audience and they'd come up here, but I won't do that to you tonight, students. This is called a dose-response curve. And a dose-response curve means that usually as you increase the dose, you increase the response. This is a little bit different. This is showing people on this axis, and this is showing the dose on this axis. And what it's saying is that for a given dose of a drug, when you go to the doctor and the doctor prescribes you something for your health, that doctor assumes you are right here. You're at this 50 percentile. You're an average person. And now, most people don't like being called average, but you are an average person when it comes to the medication that you take. And then you may have to come back and forth to that doctor saying, it's not enough, we'll come back and measure your blood pressure, or we'll do your Coumadin test, we'll see if your blood is got the right coagulation times, etc. They have to continually make adjustments. And why is that? It's because you're not the same as everybody else. You're not average. You may be below average, actually, or you could be above average in terms of the dose it takes to produce a response. But this S-shaped curve tells us to get 100% people, 100% of the people in this room, we have a large enough population in this room to talk about this, we would have to go to the dose range, and this is log scale, so it might be tenfold difference between you and somebody else, that to get everyone a therapeutic effect of whatever this drug is, you may have to up this very, very high concentration, whereas others will have that effect at a much lower concentration. So that's called an ED50, or a the 50 percentile response for this particular drug. Now, what you'll notice about this curve is something that troubles us, and that there is a red curve here as well. And that red curve says that if we're pushing you to these high doses, I mean, you could say, why don't we just give everybody a dose that'll work for everybody? And what's the answer to that? The answer is if we push you up here, now you extrapolate that dose down to the red curve, you have toxicities, you have side effects, you have problems. And so I could give personal examples of a medication that I take. I take a very, very low dose. I'm very sensitive to a lipid-lowering drug. And my side effect when I was taking it up here is I have myalgias. I have problems. But now that they've got my dose adjusted down here, I'm great. No myalgia. My golf game's better, I hope. And I have my blood lipids are where they need to be. So this is the toxic effect curve. And so we're all different. And we have to shoot for the average person. But, you know, the world is changing. As we get more and more information about how these drugs work, how they produce toxicity, then we can individualize your therapy. We may be able to do it predictively. Right now, you're a guinea pig. When you go to the doctor's office, they just give you the dose. The pharmacist provides the dose. You take the dose. You complain to the pharmacist that something's wrong or it's not working or it's too much. And then you go back to your doctor and then they adjust your dose or change it off to something else, right? You're the guinea pig. And so, but in the future, perhaps, we're going to be able to know enough about who you are, about your genetic makeup, that we can pick the right dose or get much closer the first time. And more importantly, we're going to prevent side effects and toxicities because there's a small percentage of people who have adverse effects. Some of them could be deadly, number one. And number two, some of them are certainly not reversible, so you could have a permanent injury. So that's where I'm coming from when I talk about the toxicogenomic basis of environmental health and injury and disease, thinking about how things injure you or cause disease. So that's a dose response, Pharmacology 101, and that's where we start. Another way to show that curve, we call this a quantile dose response curve, is to say here's the average dose that we give to a person for a particular medication. And this is, you probably know enough about statistics to know that that is a bell-shaped curve. That is a normal distribution. And so again, your average, you're right here, that's the dose that's being picked. But some people require a higher dose to get the response. Some people require a lower dose. Same idea as the last curve. Now, the question is why? Why are there so many differences? Someone should say genetics, but it's not purely genetics. Genetics is part of it. And so genetics of what? Well, the genetics controlling individual responses, there's many, many, many different things that determine your exposure. Does that drug get into your body? When you take a pill, does it get into your body? That's absorption. Does it distribute correctly to where it needs to go? If it's trying to work in your brain, it's a anti-seizure drug, anticonvulsant agent. Is it getting to the brain? Or is it getting metabolized in some weird way that you just basically chews it all up and it's no good by the time it gets to your brain? Or does it make a metabolite that causes some adverse reaction that you don't want? And you're going to find out, or is it excreted rapidly? Is there something about your transporters in your kidney that take it out very, very quickly? So these are the kinds of things that we have to now understand, these mechanisms, in order to give you the appropriate dose of a drug. And then someone said receptors and signaling. I think Dean Wellage, mentioned this, which is what I do. And this is a pretty complicated area, which you'll see in a little bit. But there are big differences in individuals in terms of where do the drugs act, how do they act on a particular cell, how do they convey the information from the receptor, which sits on the cell surface, to the nucleus, which controls, it's the brain of the cell, it controls the genetics of that cell. And I'm going to show you some of that in a little bit. Because I was asked to come and give a basic lecture and then show you about my research. And those are the two favorite things that I do here at UNM, is I teach and I do research. So I'm just glad to do that. Okay, so here's Genetics 101. And everyone heard of Watson and Crick, right? And Watson and Crick came up with the double helix hypothesis, and they are the ones that discovered the role of DNA in terms of coding our genes and the basic double helix structure and the nucleotides that were associated with that. ATCG are the 4 nucleotides. You can see them just here, but they are actually, it's a code. And there's millions, there's 8 billion of these nucleotides that make up the human genome. So they are put in very, very specific sequences. They are not always ATCG. They go all different kinds of patterns, and they mean something. They are a code, the genetic code. And we call that genetic code the genome. Your genome is all your genes that are in your body and how they're expressed. So the DNA is stored in this form in your chromosomes. By the way, we'll talk about your chromosomes in a few minutes. But we have 46 chromosomes. You have your diploid, you have 2 pair. You have 23 and 23. So you have 46 chromosomes, 23, and they're 23 times 2. And the central dogma of Watson and Crick, now this goes back to high school biology, right? Every one of you here that took high school, but you're trying to forget high school biology, some of you. But every one of you remembers this about DNA and about RNA. RNA is the message. And then that is what we call transcription. I'll talk about that in a minute. And then it's a slightly different code. It changes one of the bases from thymidine to uracil. And then that gets totally translated into a different kind of code consisting of amino acids. And there's 20 natural amino acids. These are predominantly taken up in your diet from the proteins and the like that we digest, that we eat. And so this is the translation part of it where we make proteins. So RNA codes for proteins, and the proteins have the function. I didn't go through the whole cell biology thing here, but you have proteins in your membranes that are structural. You have proteins that are transporters that bring things in or pump things out. You have proteins that are signaling molecules, heaven forbid. And you have proteins that are receptor molecules. So this is what we teach in pharmacy school and in medical school. And I see some of you smiling. She says, yeah, I got that. I know that. So DNA to RNA is transcription. Genetics 101. RNA to protein is translation. Now here's a word. It's called genotype and phenotype. Now you've heard these terms before, but the genotype refers to your genetic sequence, your gene structure. Now I'm going to talk a little bit about genetics, and that is, where you got your genes, and I don't mean over at at Dillard's or something. I'm talking about your genetic information came from your parents. So we'll talk about how you inherit your gene, just a little bit. Can't go too far on this. How you inherit your genes and then how that makes certain things about you unique. So the genotype is the sequence. Now I'm abbreviating SEQ because I'm going to come back and talk about seq, gene seq at the end of my talk. And phenotype is the observed state. It's actually the expressed allele. And some of these are inherited in a what we call Mendelian way. Gregor Mendel, was the guy that was doing the white peas and the different colors, the red leafed peas, and then put them together. What did he get? Sometimes he gets pink, and sometimes there was dominant and recessive alleles. So what's expressed depends upon the gene system that you're working on. And I'm going to talk about that because it's important to your immune system, which is what we're going to talk about tonight. Polymorphism means many alleles, many forms. So you have many alleles, and in our population, just to give a common something you're familiar with, if I want to take Kevin Warner's kidney and I want to transplant it into someone else in the room, in Ira's body, it's not going to work. And not because Kevin's being difficult, but because Kevin's DNA and his kidneys, the genes, are different. And Ira will reject Kevin. So that is, so we have to match. We have to match those alleles and get a good match to do a transplantation like that. We'll talk more about that later. And then lastly, you're going to hear about, you may not hear about this ever again tonight. I'll just be honest with you. But some of you in this room know about this and will hear about it, and that's SNPs. So SNPs are single nucleotide polymorphisms. And we have SNP chips. I'm going to talk about chips tonight a little bit. And I don't mean, the fish and chips. I'm talking about chips that we use in gene studies. And I've actually brought a few of them tonight. You'll see pictures on my slides later on, but I do invite you to come up later on this evening. This is an Affymetrix mRNA expression rate. The whole human genome is on this chip. That's pretty cool. And then I'll talk about some other chips when we get there a little bit later. So a SNP is basically a change in an amino acid, or change in a base. It may or may not make a change in an amino acid in the protein. So some of these things are not important. Some of these changes in the DNA base pair is not important. When we sequence you, oh, there it is, no big deal, nothing's changed. But some of them are important, and they make sometimes proteins that don't work properly. Or they knock out the coding of a protein that works at all. And I'll give you a couple of examples of that later on. The Human Genome Project. The Human Genome Project was completed, believe it or not, in the year 2000. And what was discovered from that in many, many labs all over the US and all over the world, including Los Alamos, were part of that big sequencing effort. And that effort taught us that we have about between 20 and 22,000 genes, that 20 to 22,000 genes control everything about you. And but there's a lot of junk DNA too. So the junk DNA is stuff that's not coded. This doesn't result in proteins. Nobody really knows why it's there. We know why some of it's there. But really, of all your DNA, it comes down to about 20 or 22,000 genes that determine everything about you. And I was just, I like to, when I'm teaching, I like to talk about things in the news, current events. And so this week, the Supreme Court is hearing a case. I'm going to talk about it in a minute on a breast cancer gene. And it'll determine whether or not you can patent human genes or not. At the time the human genome was done, a scientist by the name of J. Craig Venter started a company called Celera, and he of the 20,000 genes, he sequenced 6,500 of them. It's a lot. And he patented them. So the idea is that those genes might be important for diagnostic tests or doing DNA therapy, gene therapy. That may be important in the future. And he had a lot of people put money into his company. Bill Clinton, when he was president, signed an edict saying you cannot patent genes, and this company's stock went right down. Because that could be big if you own the part of the human genome. But that's going to be decided probably this week. It's being heard this week. Big, important decisions. You say, why should I care? Because, and there's two sides to these arguments, so I'm not going to really have that conversation tonight. But it's going to cost a lot of money if people can own these genes, and then basically when you want to buy diagnostic tests or if you want to do gene therapy, it'll be very expensive. So, but the patent idea is that you own it for 20 years and you make your money back from your research, and then it becomes into the public domain. So those are a lot of different ideas, things that go on in our society. So once we knew what the human genome looked like, and by the way, you got to understand when they did this, it's almost kind of silly the way they did it. They worked on these little cell lines. They were just individual cell lines. They weren't like, from individual people. They said, we're all going to work on this particular cell line and we're going to sequence it. And we're going to, because you had to everything to add up and match up. So it was very interesting. But really what we knew in 2000 was very, very limited in terms of putting these sequences together into a map. Dr. Thompson has a map on his wall of a lot of these genes and chromosomes and where they are. And it's awesome. It's amazing. It's so much information. So people started immediately looking at individuals. And we have the ability to sequence. We could take cells from you and we could do your whole genome sequence. And that costs about $100,000 to do that up until 2 years ago. And by the way, I want to say something about CSI. This is not CSI. What you see, and everybody comes to us doing, I want to be like CSI. And toxicology, that's forensics, but it's not really the same. They're not really sequencing. What they are doing is they're just cutting up your DNA and looking at patterns. So you see them with these gels with the lines and there's markers. So we can tell who's, they use these in paternity suits, so who begat who, that sort of thing. But so there are a lot of things you can do with this, but they're not actually sequencing all of the genes on CSI, just so you know. So genome-wide, they started looking for, okay, I have a group of 100 people here. I'm actually working on this. I got 100 different cell lines that I made from women who had reduction mammoplasties that I was studying for breast cancer. And then I'm looking for their responses to different environmental chemicals which are known to cause breast cancer. And so I thought, well, that's great. I got 100. And then you can look at who's a responder, who's not a responder, and then you can do sequencing, and you can think, or sort of do sequencing. You can do a variety of experiments. We'll leave it at that for now. But people would do the sequencing, and then they try to do an association study. We call them GWAS, Genome-Wide Association Study. And when you do this with a lot of people, you can't do it with 100. You have to, tens of thousands or more. Then you find certain genes are associated with certain diseases. I'm going to talk about one in a minute for breast cancer, okay? But there are not very many of those genes around. It turns out most diseases are very complex. There are many different genes that control almost anything, any phenotype we observe. Let me give you an example. One of the most complex phenotypes is your height. What determines how tall you are? There's no height gene. There are dozens of different genes that control that. Now you can start imagining, knowing what affects, if I feed my kid, they get bigger. If I don't feed them, they don't get smaller. But nutrition has played a huge role in this. There's a lot of other things. But tall kids, like our basketball coaches, their kids are tall. They played basketball and their kids are tall. And so they're passing on some genes that are contributing, but it's not a single height gene, or believe me, the Lobos would have it. They would be out there cloning those genes and using it. So it's many genes, polygenetic. And so this is true about all diseases that we have, actually. There are a few high penetrance genes. I'm talking like a geneticist. I love it. So there are a few high penetrance genes. That means if you got it, you're going to get it. Okay? If you have this gene, you're going to get it. So the one that's scary is this BRCA1 and BRCA2, that if you have that gene, it is so highly penetrant. So many genes are affected by lots of other things. This gene, if you have it, you're going to get breast cancer. So we screen for it. And that's what the fight right now with Myriad Genetics. This is the case before the Supreme Court this week. Myriad Genetics of Seattle is patented BRCA1 and BRCA2. And those of you who've had the test know that it's cost thousands and thousands of dollars to run this test. And so, is that good? If you know if you've got the gene, you can make a decision, make an intelligent decision about whether or not you want to do something about it. Last month in the journal Nature, so this is the BRCA gene's been known for about 10 years. It was discovered through the GWAS type approach, but there aren't many genes that fall into that. Last week in Nature, there was a study. It's called the Cancer, and I forget their specific name, Cancer Atlas Committee, I believe what's called, not committee. Cancer Atlas Project. And they published 47 new breast cancer associated genes. That study involved 200,000 women. So we got to have big numbers because there's a lot of genes. Now, these are not high penetrance genes, and these are not genes that are purely rely on genetics. In fact, most of the diseases that we have occur because of an interaction of a gene in some environmental signal. And we call that a G times E, gene times environment interaction. That's what we work on. It took a long time to tell you what basically we're working on, but that's basically what we work on, gene-environment interactions. But it's no single gene for most things. So I put it's G times E times G times E times G. You can do this any way you want. You can make a matrix and put G's here and E's over here. And, you know, the point is that these are very time-dependent, temporal. They're very temporally dependent. So when it happens is very, very critical. So we now know developmental exposures. That's why we're so careful when women are pregnant. Make sure they don't have alcohol. Make sure they get the right vitamins. Make sure they're protected. They don't get radiation from a scan, because the developing fetus is super sensitive to environmental effects. The genes, the tissues are so, they're going through so much change and growth and development. We have to protect them from these outside signals, these outside events that are damaging. So that's what we do. We're also doing something else, which unfortunately to us is even a lot more complicated. Not only do you have a gene, a series of genes, the genome to think about, but it turns out that genes are controlled by proteins that wrap around the DNA called histone proteins. And these proteins that coat the DNA are imprinted by biochemical reactions, including methylation, acetylation, others. It is really complicated. And we're just trying to start figuring it out by doing the kinds of studies we do. And I'm going to pass a cup at the end of the plate at the end, if it's okay, because these studies are super expensive. And so I just want to thank Nunzio and Cheryl and DeSantis, because endowing my chair in pharmacogenomics, it's forced me to pursue this work. And I don't really have the funding to do a full project, so we keep kind of working away on it. And it's so cool that the technology has changing so rapidly that we can now afford to do. So I told you a few minutes ago, a human genome study, sequencing your genome, costs about $100,000, and I'm hoping next week we can do it for about $2,000. It's really changed in how this is going on. So that's technology, and then we can actually ask questions. All right, I'm going to go through this quickly because I really wanted to do, I think you guys could relate to this. I'm trying to explain to you that we're different in the way we react to chemicals. And one of the best ways to talk about that is drug allergic reactions. How many of you are allergic to a drug in the room? That's a bunch of you, okay? And I'm not going to call out, I usually do this in class. Or I say, how many of you are allergic to a food? I know my sister is. Almost killed her once with peanuts. Okay, you're allergic to foods, and so fewer. And then I'll go around my classroom in my immunology, what are you allergic to? And someone says, watermelon. And I was like, I never heard that. But there are people allergic to watermelon, as it turns out. So lots of stuff. Why are you allergic? So I'm going to summarize now. I don't want to take too long this evening. But we have common experiences. We know that people have drug allergies. How many people have seasonal allergies, hay fevers? You know, a lot of people do, okay? So there are common allergens and there are uncommon allergens, okay? And what does it require? It requires exposure. You're not going to be allergic to something you've never been exposed. Now, you may say, I never took penicillin before. You may not know that you had been exposed to penicillin because, unfortunately, they use it on the ranches and the milk and everything else. In fact, we have to screen the milk for antibiotics to make sure that people, who are sensitive don't get it, but B, we don't sensitize people who would be sensitive, among other things. So there's other things in foods that we get exposed to. But you must be exposed to be sensitized, and then you must be exposed to get the symptoms, to have your allergic reaction. And so most of you know you're allergic because you are a guinea pig. You were exposed, and then you got a reaction, and then you reported it, and we said, okay, you go to your drug record, and they say, I'm allergic to sulfonamides. So every time I go in, I have to tell them that. You'd think they know that. I've told them that for how many years, but the medical records aren't really up to where they need to be just yet. But so anyway, you tell it to everybody you go to, and they write it down, and they try not to give it to you. Common, we don't have it around here because of our altitude. Poison ivy, poison oak is a sensitizer, is a contact sensitizer. If you rub it on your skin, the leaf, there's actually chemicals in the leaves that come off. They bind to proteins in your skin, and they will sensitize you. And then if you do it again, you'll get rash, blistering, itchiness, erythema, all these things, reddening. So those things will happen. This is common. 80 to 90% of people who are exposed to poison ivy will respond. Okay, so it's not uncommon. So you avoid exposure, right? Or you suffer the consequences. But a drug like penicillin, which is one of the most common drugs that people are allergic to, even that, it's between 5 and 10% of people. So in this room, there's probably 4 or 5 people. How many people are allergic to penicillin? Okay, 1, 2, 3, 4, 5, 6. So, I mean, that's kind of statistics, kind of the odds. But we are going to try and make sure that you don't get penicillin, and that's something we have a lot of experience with. The reactions you get are quite varied, actually, because I didn't ask you, do you have anaphylaxis? Do you have hypotension? Do you have trouble breathing? What is it that when you get exposed? Is it immediate? Is it delayed? Et cetera, et cetera. There's 4 different types of reactions. I won't go through this now, but you can have a lot of different kinds of reactions immunologically. Being sensitive can mean different things to different people. But we are starting to understand the genes that are responsible for these sensitivities. We're not there yet, but we hope to have it on your DNA drug card someday. When you scan it, they have all your drugs, you have your genotype, your DNA genome on your card, and we now, with the computers and all the fancy stuff we can figure it out, I hope, maybe 10 or 20 years from now. So we can screen. So these are common things that people are allergic to. I was going to do the survey then, but I won't take the time now. A lot of different things, foods. Okay, here is a poor guy who has Stevens-Johnson syndrome, and it's also called toxic epidermal necrolysis. Necrolysis means your skin is sloughing off and dying, and it can be fatal. And I've worked on cases for people that have this, and I've worked with companies developing new drugs to say, oops, we got this problem in a small percentage of people, but the problem is for this new drug that you're developing, you just can't throw it out there in the general population and say, oops, a bunch of people got sick. So it would be nice if there was a way to say, these are the people who will benefit from the drug. These are the people over here, probably a small group, who we should not give that drug to. That's the thought. So it turns out that, and you can barely see it up here, this is associated with one of your genetic markers on your immune cells, and it's called HLA-B1502. So now we can screen people. Do you have that particular allele? And if you do, we won't give you that drug. And so I was going to go give you an immunology lecture, but I don't think I'll take the time today. But I talked to you about cells that are immune cells that are in your blood. And when we want to do, I've already talked about Kevin's kidney. When we want to know what his genes are, we have genes that are called histocompatibility genes. Those are tissue genes. They are expressed on every cell in your body, except the red cells, expressed on every nucleated cell in your body, and they are class 1 and class 2. And they're called MHC, major histocompatibility. These are the genes we match. And the ones that are the class 2 genes here are expressed on the surface of these immune cells, and they're involved in recognizing and presenting antigens to your responder white blood cells. I'm trying to keep it simple if I can. And so they are the ones responsible for that Stevens-Johnson syndrome. Okay? If you have HLA-B22, then here are the different HLA genes. HLA-B right here. So if you have that B1502, you've got something there. Now you'll notice something about this. There are 2 sets of these. There's one from your mom, maternal, and there's one, class 2s are up here, maternal and paternal. So these are genes that are called codominant. So it's not like Mendelian where one's dominant over the other. This is, you get all of them. So you can imagine the number of how unique you are relative to these MHC genes. I haven't told you how many there are, but there are hundreds. So when you take all the different combinations, everyone in this room is pretty unique, unless you've married in your family or something, and somebody said you're from North Carolina at the Kingston Trio concert the other night. So I don't know. I don't want to besmirch anybody. So this is a brave new world that we're in, and this is my previous slide said coming to a pharmacy near you. These products have already been launched. They tried to launch them in the in Walgreens and CVS and the like last year, and physicians said, please don't. American Medical Association said, please don't. Why is that? These are testing for candidate genes that are associated with different phenotypes, different, whatever it might be, a drug reaction. It's a lot of different things. And so this is very eloquent, actually. And you get a little box. I just did this, and Dr. Thompson and I shared it. Well, we didn't share our samples, but we each participated in this. And by the way, it was $200 when I did it. It's $99 now. So it's getting cheaper. But they will screen for thousands of genes. They're not going to sequence you, but they have a way to look for thousands of genes of interest. And it's your spit kit. I don't know if you can read that or not, but you do. You really get it. They tell you to work your cheeks, and you fill it up. And there's enough of your buccal cells that come off of your cheek and the like that's enough DNA that they can then test you. You mail it in, and 2 weeks later, they send you back information about yourself. Some of it you didn't know, some of it you may not want to know, but it's there. And so this is called 23andMe. Why is it called 23andMe? Because you have 23 unique chromosomes, right? You have 2 pairs of them, so that's where that came from. And basically, you could do ancestry studies if you wanted to. There's markers for that. But we use them for health studies, and we use them for research. So we're part of a huge cohort. And the reason they dropped their price to $99 is they're trying to get 1 million people by the end of this year to have done this, because then they have power in their statistical analysis. I told you the studies that were just done and published in Nature were 200,000 people. They're going to have 1 million people in their database. So that's powerful when you can start looking at all these different genes. And it's kind of interesting. I was actually going to put this out there for the students. Ann Wojcinski, who's the CEO of 23andMe, Mark Zuckerberg, who's the Facebook guy, they created a new, it's called Life Sciences Breakthrough Prize. They announced these last month. They gave 10 of them, and they're $3 million each. They're double the Nobel Prize. And so that's pretty good incentive for people who want to work on something really cool and interesting. And they're going to give that every year. Okay, so here's one of my results. And this is Scott Burchiel's result, my genetic data. And this is about a gene that's called HLA-B57, another one of these HLA human leukocyte genes, 5701, where there's and in this particular case, this is a gene, it says right here, it's a drug called abacavir. Abacavir is a drug used to treat HIV, and it talks about 8% of the people treated with abacavir develop a hypersensitivity like Stevens-Johnson syndrome. not a trivial thing. It's others as well, but you can have Stevens-Johnson. So they now screen people. Everyone who is hypersensitive to abacavir has a particular immune marker, and they say that now that they screen people, it's good practice, it's considered best practice to screen people before you put them on that. And the prevalence, though, is very different, as you might think, in different populations. 5 to 7% of people with European ancestry carry the marker. In India, 20 to 50% carry the marker. So it's very interesting, and it's virtually absent in Japan. So it's very, very different from one group to another. So I put this in actually for one of my students who works at Sandia, but she couldn't make it tonight, but Larry's here from Sandia Clevenger, who works in this area. I mentioned beryllium earlier. Beryllium is also a sensitivity, a hypersensitivity disease. And it is got a marker now where we can actually screen people to say whether you would develop chronic beryllium disease. And it forms granulomas in your lungs, and it might take 10 years to present, and it might take 20 years, but it will kill you. There's no cure. It's maybe a 30-year disease, and maybe shorter, but there's nothing we can do. Even people working at secretaries, working in buildings where they're not actually handling beryllium. It's used in the nuclear industry because it's a very strong metal alloy that's very light. The Air Force commissioned a National Academy of Science committee that I worked on, and we wrote them a report about this. And they said, what is a safe dose, or a TLD as they call it, what is a safe level of exposure to beryllium? And we said 0. And that just drove them crazy. Because it's like saying, if you're allergic to penicillin, what is a safe dose of penicillin for you? So people have to deal with some of those kinds of things. All right. I often tell my students, we've been sort of skimming along the top, and then we're going to dive down a little bit, touch the bottom, and see what's down there. And everything I've told you about up to now is stuff that I'm involved in teaching, but I haven't published any papers in that area. So I'm going to just go through a few things that we do, and I'm going to watch your eyes. And when people start walking out of the back, I'll probably shorten it up a little bit. But here we go. We're going to dive down a little bit, and I can make a lot of comments about that, but I won't. So I work on a class of environmental chemicals called polycyclic aromatic hydrocarbons, PAHs. And I actually work on several of these because there's thousands of them, even in tobacco smoke, and in car exhaust, and in standing in front of your wood-burning fireplace. You're getting exposed to these PAHs. They're formed by combustion of fossil fuels. And the hotter the flame, the longer it goes, the bigger. They look like chicken wire. That's my best way to describe a PAH. They look like chicken wire. And there's like 5 fused, 6 fused rings. But to get all those benzene rings fused takes high temperatures. And so the bigger they are, the less prevalent they are. But unfortunately, the bigger they are, the more toxic they are. They cause cancer, and they suppress your immune system. And that's what we've worked on for about 25 years. So I'd like to tell you just a little bit about it. And they require metabolic activation by enzymes in your liver and elsewhere called P450s, and they form reactive metabolites. When they're reactive, they bind to your DNA, and the way they cause cancer is they are mutagens. They mutate your DNA. Your DNA, when your cells replicate, they make a mistake because there's some big bulky PAH molecule bound to one of those bases. It's called an adduct, and it messes up. It puts the wrong base in there, and that becomes, in many genes, a cancer gene. So an oncogene. So that's the kind of stuff we do. The same thing happens in the immune cell. But in the immune cell, we have sensing mechanisms called P53, and P53 is a sensor in the cell to pick up these adducts. And it says, if you activate your P53, the cell commits suicide. It doesn't want to replicate. It undergoes what's called apoptosis. So it's very interesting at low levels of exposure, P53 can be activated, and maybe the cell doesn't commit suicide. The other thing I want to talk about is oxidative stress, something I've tried to explain to my family and friends for years. What is oxidative stress? I know we're under stress, But oxidative stress is not an easy concept to explain to people. So I'm going to show you a first part of the story with June's work, June Gao, who's in the audience. June did all of these studies and published 4 or 5 nice papers on this. And so here's a chemical called DMBA. When we use it in mice, it causes breast cancer. We used it for many years. That's why I got interested in it originally. It gets bioactivated by this P450 CYP1B1, forms an epoxide, and then in the presence of an epoxide hydrolase enzyme, it forms a diol, and then it gets hit again by this same enzyme, CYP1B1, and forms something called a diol epoxide. And it's reactive, and it binds to DNA, and it can mutate the DNA, but it can also be sensed. This is all June's work. It can be sensed by these sensors, activate the P53, cause the cell to die, or just at least to stop proliferating. So June's work, I'm not going to show you any of your data, June, but June's work, we use, my daughter thinks this is like a crazy concept, that we engineer mice. We make mice where we knock out genes. And so we use these mice that were CYP1B1 null. They knocked them out. And then we proved that without this activation, nothing happens. We proved that when we use epoxide hydrolase knockout mice, nothing happened. So we know it's part of this sequence of activation. And then we use P53 knockout mice. Now, we don't make most of these. We can get them from the NIH mutant mouse repository. And we can just order them up and breed them up, and we got happy little mice. Although these guys are pretty sick. So P53 mice, so we've gone through this whole cycle, every one of these, and we've shown that it works. Now, here's an example for those of you who smoke, and I hope we don't. This is what happens with those PAHs when you smoke. And this is relating to a study I'm doing with Columbia University now. And so these are the PAH adducts. So that means these are those bulky things bound to the DNA. We can detect them a variety of ways, which I don't want to go into. But this is looking at the number of cigarettes per day you claim you smoke, and then you report. And then we take your blood cells, your white blood cells, and we look for the number of PAH adducts, like benzpyrene, on your DNA. This is not good, okay? I think we've established that. But look at the range. Okay, well, see, so this is someone smoking 40 cigarettes a day. Is that possible? I guess it is. But the average person smoking around 20, in this group, smoking about 28. Look at the range. Some people, there's a huge number of adducts, and some are very, very low. Why is that? Well, 2 general reasons. Number 1, are you activating it to form the adduct to begin with? But the other reason is these adducts get repaired, DNA repaired. So there's many different pathways that fix it. You know, you have 100,000 mutations in your body every day, just through the normal cells proliferating and doing what they're going to do in your body. The cell has to have very complicated pathways to fix those mistakes. And so the DNA repair pathways. So we're starting to get very interested in those. Okay, well, I know I'm not going to get to talk about all these genes, but these are the genes that people are interested in. My colleagues at Columbia, this is from Regina Santella that I work with at Columbia. These are genes that we know what they do. Some of them are DNA repair genes, some of them are oxidative stress genes, but what I want to point out, I was hoping to get you to sort of get to know some of these names, but let's not worry about that. Look at over here in this column, it says the variant frequency. So what percentage of people, now this is going to depend on who you're studying, obviously. European, African, ancestry, Asian, it's going to differ a lot. But this is her table, her numbers. And so some of them are pretty prevalent. Some of these polymorphisms, some of these changes are pretty prevalent. So I would just say that has to do with DNA repair and DNA repair genes. Well, some of the genes that we're interested in are these oxidative stress genes. So I'm going to take a stab at explaining what that means to you and show you a little bit. But oxidative stress is something that's happening in all of us all the time. It's a function of your mitochondria that are making ATP, which you need to live, which you need to run. But that process generates reactive oxygens, among other things. It's always going on. It's responsible for your aging. It's responsible for your skin getting kind of wrinkly. A lot of that's from the sun as well, which causes oxidative stress. So aging is an oxidative stress phenomenon. Your cell has ability to resist that stress. There's a variety of mechanisms. I'll just talk about one. And it's called glutathione. And when you see this SH, it means this is sulfhydryl, which means it will accept the oxygen and protect your other molecules from being oxidized. You know, when you park your car out in the sun and you see what happens to the paint, it gets oxidized. Well, that's happening in your body. Oxidative stress is happening in your body. So we have lots of different pathways to fix this, to keep this in check. Glutathione is one of the main ones, and it's present at very high millimolar concentrations in your cells. And the interesting thing is, if I'd gone back to that slide, you'd see a lot of these GST genes, which are glutathione-related genes, which are protecting you. So if you don't have the ability to resist these stressors, you're going to have problems. You're going to be more sensitive to these chemicals. Cancer, immune suppression, for example. Now, there are things that in our food, so a lot of people advocate, you know, we should be eating healthier, which my wife advocates that to me all the time. And vitamin C and vitamin E are antioxidant vitamins, so we should supplement. We should take those vitamins. There are things in plants. One group's called flavonoids. These are all collectively called polyphenols. Flavonoids in plants, like quercetin is an example of one. And so now, you know, you go to these herbal stores and say, take this, take that. This is good for you. Some of it's true. Some of it's unproven. In wine, there's an antioxidant called resveratrol. And so people are selling that in the health food stores and the like. So these are the kinds of things your cell is trying to, you can do, you can protect. Your genetics will determine what you have. In your cell, there's a lot of other genes. Now this gets to the good part, where we're going to talk about antioxidant response elements and genes that respond to oxidative stress. So let me just run this slide by you. This is the signaling slide that everyone's been waiting for, which I am famous in our department for throwing these kinds of slides, and this is very simplified. So this is just a slide showing, this is going to be a T cell, for example. I'm going to talk about T lymphocytes for the rest of my talk. And this is the antigen receptors, but I'm not going to go through this whole slide, believe me. But let me just say that there are these reactive oxygen species that occur in your cells. They come from a lot of different places, mitochondria, lots of different things. And there's 3 different ones that I'm going to show you an example in a minute where they come from. But these reactive oxygen species, and so I'm going to generate them with my favorite chemicals, the PAHs, the benzpyrene quinones. I'm going to generate them with that, and I'll show you a slide in that in a minute. And then there's sensors in the cell that says, wait a minute, we got to get rid of you. There's glutathione and other things I've talked about, but when those buffering capacities get exceeded, we turn on genes which are called antioxidant response genes, and they are generated by these antioxidant response elements, which are on the DNA. So how that works, and one of our colleagues in Arizona is one of the top people in this field of NRF2, and NRF is a transcription factor. It's usually bound in the cytoplasm of the cell. So this is a cell, this is the nucleus, this is the cytoplasm. So these things are happening out here, the signaling. And these reactive oxygens, when they bind to this protein KEAP1, KEAP1 is a sulfhydryl-rich protein, and those sulfhydryls are critical to bind up this transcription factor. When they get oxidized, the NRF gets released, and the NRF goes into the nucleus and turns on these antioxidant response elements, and it makes enzymes like NQO1, N-quino-oxidoreductase. So these are quinones up here that we're feeding them. It's saying, we don't like you, we're going to get rid of you, we're going to metabolize. There's a whole bunch of these. In fact, there's a battery of genes. So here's our friend, 1,6-BPQ, and BPQ is one of these chemicals that's a PAH, and Matt did a little bit of this work. The UV there, you can activate, you take your PAH out in the light and the UV will activate it. And so you form a quinone. And then the quinone undergoes, without enzymes or anything, this thing called redox cycling. Round and round and round it goes. It doesn't ever stop. It forms a semiquinone radical and it forms this dihydrodiol. And every time it does it, it generates this stuff called superoxide anion. And so superoxide is one of those reactive oxygens. And then in the presence of an enzyme called superoxide dismutase, SOD, it makes hydrogen peroxide. Now you think hydrogen peroxide is not that bad because you have it in your medicine cabinet at home. You pour it on wound, whatever. In your cell, it's nasty. Because in your cell, you have iron present, and it does something called Fenton reaction, forms a hydroxyl radical, which is nasty, nasty oxidative stress. So these are three superoxide, anion, hydrogen peroxide, and hydroxyl radical, or three ROS. Now, Dr. Fung, who's in the back of the room, works on reactive nitrogens as well. So some of these can actually trigger another set of reactive species, which are also nasty. So all that being said, let's talk about genomics. And so if we take cells and we have them growing in a tissue culture plate, we don't always do it this way, but if we do, we expose them to a quinone, and then we extract the cells. We extract the RNA. I want to know what genes are turned on when I expose them to the quinone. And so we take them, and then we put them on these little gene chips, okay? And then we read them out, and I've really simplified it because we generated about 1,200 genes out of the 22,000. But I'm just going to give you this is like Dave Letterman. This is my top 20 or top 30. And what I want to just say is that on this gene list are genes that we know what they do, and that's what's kind of cool because they're involved in the metabolism. Here's the NQL1. They're involved in the metabolism to protect the cell. There's oxidative stress genes that are in here. Here's a glutamate cysteine ligase, which is a glutathione synthesis gene. We can take this information and we can understand what's happening in the cell, and that's pretty exciting. And secondly, we generate a bunch of things which we have no clue. And so when you go fishing, sometimes you catch fish, you don't know what they are, and then you got to go figure it out. So then they allow us to generate hypotheses about, well, why is that up? What does that relate to? And that's what we do. And sometimes you have to do something like this, gene pathway analysis, where you don't know, but there's been a lot of papers published. So you feed your information into a network diagram, and it tells you, these are your nodes. These are the things that, this is not my data, by the way. So there are nodes, and they're all connected to your genes through these different pathways. So this is the field of bioinformatics. And bioinformatics is a huge, for students, this is a huge area for people to work in now, to take data and to be able to analyze it, because the data sets are huge. When I run one of these chips, I generate 55,000 pieces of data. The next this was a few years ago. The next generation of these chips, when I run one chip, I generate 500,000 pieces of data. Okay? So that's what that's about. I'm actually going to stop because I think we've gone long enough. I wanted to tell you about what we're doing on my new research, which is arsenic and PAH interaction. Arsenic is a huge worldwide public health problem. It's just now being appreciated. This paper was published in The Lancet by my colleagues at Columbia, which we're now working with, Columbia University. And I just want to read you this statement. The statement is, based on the risk estimates, these are people that have high arsenic. Now, we have high arsenic in New Mexico. We used to have the highest municipal arsenic drinking water levels in the United States, but we put in an arsenic treatment plant. Then we brought in the Chamba River, and we mixed it with snowmelt water. We don't have the problem today. But in wells on Indian country up in Navajo, huge levels. So there's a lot of ranch wells, huge arsenic. And arsenic causes bladder cancer, skin cancer, other cancers, and also suppresses the immune system, which is what we're looking at. So based on this, a study done in Bangladesh, which is we're starting to do a study in Bangladesh on immune function. It's never been looked at. And based on the estimates they find, 23.5% of all deaths are associated with arsenic in Bangladesh. Now that means arsenic is contributing to the disease. They were at higher risk. It's heart disease, it's cancer, it's a lot of things. But 23.5% of all deaths were associated with arsenic. And we have populations or pockets in the United States which also have risk. One of the things that we have discovered is that so I started the talk by talking about sensitivity, drug allergies. But I'm now telling you that we're discovering that there are people who are sensitive to arsenic. not in the sense that they have, allergic reaction, but in the sense that their T cells are really sensitive to suppression. And that's what we're working on. And so we've now done a study. We've done 217 as of today, donors, and we have identified people who are responsive to arsenic at levels that are 1,000 times less than what EPA says is safe to drink. So we would like to know who those people are. We would like to find out if that really is a health hazard or not. We would really like, but we have to do this. All my studies are done in vitro, where I add the arsenic to your cells. So people come, they donate blood, we put them down and we study their cells. But we won't put them down. We put their cells down. We keep track. We put the data down. No, please donate. We need donors. We don't put them down. So anyway, but we're finding only maybe about 10%. Maybe that's good. But if you think about the allergy sensitivity thing, that's still a fair number of people around the world. So I was going to take you through and tell you about our really cool stuff. This is the data I'm going to give at my conference in 2 weeks. So I guess I'll just have to have you join me in Honolulu in 2 weeks if you'd like to see this data. I'm going to close with this other neat device. I told you we can now do sequencing for $2,000, and this is something that just came out. It's a next-gen sequencer. It's called an ion torrent. This is called a proton torrent. And these chips, when I say we have chips, this is a chip with 10 million wells on it. So I'm going to get 10 million pieces. Remember, I went from 50,000 to 500,000, 10 million on this. That's a lot of data to have to sort out. You got to use your iPad or something. And then the newest machine, which we were one of the first institutions to get one of these. It's just a little bit bigger. Okay, so here's this one. You guys you can look at these and play with them if you want. Come up. But see, this one's a little bit bigger. This is the proton torrent, and the proton torrent chip has 100 million wells. So I'm going to get 100 million data points from every time I run a sample. So please donate to my lab, and it's actually pretty exciting if you think about it. I'm not going to talk about our chipping, which is the chromatin stuff. That's where we're going with it. But I want to thank everybody again, supportive UNM and HSC administration. It's the environment that brings researchers in. It's the environment that keeps researchers and teachers here. And it's been a great environment at UNM. And I thank the provost and all of John, all the people and the dean, everyone who fights to keep UNM at the top of its game in research, not only basketball. And I thank you for the investments in the future, because a lot of the work that we do is expensive and we need money. But we need, most of these projects we've had through seed money, and the university is very good about providing seed money to get you started. And my endowment has allowed me to do a lot of extra things. But then we can get the data to write the grant to do the work. The buildings we have, the facilities we have, I wish I could show you our new buildings and facilities. They're spectacular. And core labs are very expensive. We're using a lot of technology. I didn't get a chance to show you flow cytometry, genomics, but these are some of the people that are involved, research centers. So thank you. And then finally, to acknowledge our current lab members, and they're here, and I'd actually like those of you here to stand up. The number one is Ferdine, who's on vacation but came back today. And Peace is here. Peace, if you'd stand up. And Shay is here. Shay's an undergraduate working in the lab. And Deb McKenzie. So thank you for coming. And I've already mentioned several other people that are former members that we still talk to them. But thank you for coming. And then my collaborators, Jim Liu, Lori Hudson, Todd, I've already mentioned, is the one that's been sort of my conscience on genomics. Like, you've got the chair, you need to be working in this area. And I'm like, okay, we'll do something. So we've been working on it. Ellen's not here tonight, but Ellen's got us involved in the T-cell story. It's pretty new stuff. Mary Walker's been helping us. Matt's here. Matt took over the Environmental Health Signature Program. And then Pope Mosley, I think he's in Denmark. I don't know where he is. But he was my partner with the NIEHS Center for many years. Dean Wellage, thank you very much for supportive, and my chair, Linda Felton, thank you very much. Bill Halley, my former dean, thought he could come, but he couldn't come. He lives in Phoenix now. I thank my collaborators. Of course, I thank my funding sources, NIEHS, whatever. And I just thought this is something cool that we have a program now for doing our Translational Research Center, which is called Vivo. And Vivo allows you to, we'll go back and look at your publications over the last X numbers of years, and then they'll show you your network, your author network. So, of course, they ran it on me, so I'm in the middle. But the bigger the ball, the more the papers. So there's Ferdine, big ball, and then a lot of you that are in the room. You're around this circle, and I thank you very much for that. So I want to thank family, friends, big group from Del Norte Baptist. Thank you for coming tonight. I really appreciate that. And Chris, thank you for coming and asking me what a gnome is. And I want to thank my wife, especially Judy. Thanks a lot.
Yeah, I'll stand over
stand
here.
over, okay.
Thank you. So, let's see. Where's the, it's right here. Thank you, Scott. That was wonderful. I understood the first 2 slides and the last 2 slides. So, but I want to thank you first of all for your presentation tonight, and I want to present you with a citation in here. I'm gonna try to read a couple of things in here. It's a long citation, and it's signed by John, our Vice President for Research. But the citation is recognizing your extraordinary work in what you have contributed to the University of New Mexico. Personally, I would like to thank you for your effort in your teaching and your research that spanned 36 years at the University of New Mexico. And I look forward to celebrating more of your achievement in years to come. I also would like to thank everyone who's attended tonight's lecture. Special thanks to the members of the Research Policy Committee who have offered their time to select a lecturer, and to the staff in the Office of the Vice President for Research for planning this splendid event. I'm going to read a couple of things in here from the citation. Scott Burchiel is an outstanding researcher, educator, leader, and administrator at the University of New Mexico. Not many people can put all of these things next to each other. And he joined UNM, as we heard, in 1977 as the National Cancer Institute NRSA Fellow, became a faculty in the College of Pharmacy in 1978. I'm going to skip to the last part because it's a long citation, but the University of New Mexico proudly congratulates Scott Burchiel as UNM's 58th annual research lecturer. Thank you so much, and I invite you to join us next door for coffee and dessert. Thank you.
Can you read it for me?
All right.